Hey guys, it's Mr. Kenny. I'm back with a little bit about DNA replication. Now you realize that in order for us to actually grow, our cells have to replicate themselves through a process of mitosis. And if you remember, during interphase, one key thing happens during actually the S phase of interphase, and that is the synthesis of DNA, where DNA copies itself. And it's not a simple process, but let's go through a little bit. All right. When we talk about replication, the very first step is the DNA has to be unwound. You know it's in this double helix form, and it has to be unwound, and it's done by an enzyme. The enzyme is called helicase. Now remember, anytime you see the ASE on the end of a word, it means it is an enzyme. And this process of DNA replication involves dozens and dozens of enzymes. The second step is building the daughter strand. So once you get it unwound, you have to make the daughter strand, the complementary strand of DNA. And this is involved with DNA polymerase 3. What it does, it comes on and it puts the appropriate nitrogen bases. So C's with G's, A's with T's, G's with C's, etc. Now, when they add it, there's a unique principle about this, that it can only add nucleotides to the 3' end. Now, just a refresher, if you look at this sugar down here, here's the 3' end, this is the 5' end, so it goes 1, 2, 3, 4, 5. You can only add going in this direction, and if you remember, it's anti-parallel, so it can only add at the 3' end, either this way or that way. Now, it needs some type of starter nucleotide in order to go. It can't just bind. So just remember that the strand only grows from 5' to 3'. All right. Now, when we talk about it, whenever it unzips and you got this helicase splitting it apart, the DNA polymerases go along adding things. You're going to have two strands. You're going to have a leading strand and a lagging strand. Now, from this direction down here, you're going at the 5' to 3' way, then that is going to work. And that's going to be a leading strand. It's going to just go boom, 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 boom pretty quickly and do it. But if on the other side of the replication fork, realize it's going in the wrong way. It's going from 3 to 5'. It will not work this way. So what happens is we actually have to go in the other direction, and it goes in these little segments in the other direction, because that's the limit to polymerase 3, that it can only bind to the 3' end of the DNA. So that makes us have to go the other way, and that creates what's called the lagging strand, or Okazaki fragments. Okazaki fragments was discovered by this Japanese scientist. And Okazaki fragments are not going to happen as quickly. The leading strand is at the bottom, all right, and it's the one that's going pretty smoothly. It works just like you would think. The lagging strand is the one at the top. It's going to kind of lag behind in its production. Now, and when it gets done, there's going to be regions between these lagging strands that are not put together. And you have an enzyme called ligase that comes in and fills these spots in and makes a continuous DNA strand. All right. Now, if we just do a refresher, you got the replication fork and the replication bubble. You know, you got your DNA up here. It's kind of like a zipper. And the helicase comes and unzips it. But it doesn't unzip it just and start in one direction. It actually goes in both directions, all right, which makes the process quicker. DNA polymerase 3 is going in one direction, doing the leading strand, does pretty well. The lagging strand is going in the other direction, right, little bits at a time. And if you look, the forks are actually going in both directions. So if we look at this, we actually have a leading strand on top, right? Here's a leading strand up here and a leading strand down here. And this is the lagging strands are going to be on this side and the lagging strands on this side, and they're going to be working in both directions. Eventually, the replication forks will meet themselves. There'll be more than one replication fork, and the process is pretty quick. All right. Now, the limits to DNA polymerase 3 is that, remember, it can only bind to the 3' end of existing DNA. So how does it start? It also needs what's called an RNA primer. All right, this primer is called primase. Is built by primase, I should say. Primase is the enzyme that binds the RNA. So whenever you're making the new DNA strand, you actually make a little bit of RNA to start with, and that's what the DNA is hooked onto. It serves as a starter. All right, and the same thing has to happen in the lagging strands as well. All right, and it keeps on going. Now, when it gets done, if you just stop there, you would have little segments of RNA inside your DNA. Well, we know that can't happen. Well, you have another enzyme called DNA polymerase 1, and what it does is it comes in and it changes the RNA primer to DNA nucleotides, and it makes a continuous DNA molecule. Now, this is what makes this happen, allows us to have what we expect to have happen. But by doing this, we often have what's called chromosome erosion, or part of the chromosome is lost, because if we come in and we replace part of this RNA code, then some of that information that is there is going to be lost as nonsense. And chromosomes will get shorter over time through each replication. I think it's a limit of about 50 replications for every cell that can occur. But what we do is we have things called telomerase. Telomerase controls telomeres, and what they do is it comes in and puts a segment like TTAGGG into the space where it's replacing that RNA, and this leads to making it back into DNA. Okay. Now, if we talk about a replication fork just real quickly, so if we got replication. Just as a summary, you got helicase unzipping it, right? The primase is coming in making the RNA starter. DNA polymerase 3 is the one that's making the nucleotides and laying it down. You got the Okazaki fragments that are in the lagging strand, and they're being joined together by ligase. Polymerase 1 is going back in and substituting in for the RNA back to DNA and making them continuous. So that's kind of the entire process summarized. And, you know, DNA polymerases are amazing. DNA polymerase 3 actually does about 1,000 bases per second, so it's pretty quick. It's called a DNA builder. And DNA polymerase 1 does about 20 bases per second. It kind of is the editing part. It takes away some of the errors and stuff like that, so it's a little bit slower. DNA polymerase 1 proofreads it. Just to give you an example, there's about 1 in every 10,000 nitrogen base errors until DNA polymerase 1 comes through, and then it goes to about 1 in every 100 million bases or error. It's amazing if you think about how fast it's replicated. And just to give you an example of how fast it's replicated, E. coli can replicate all of it, a single chromosome in less than an hour. Humans, we have 6 billion bases, and when we divide into, it takes us a few hours to copy all those over. But we still have very few errors, less than 1 error per 100 million bases. That's pretty daggone good. All right, now the last thing is, how does all this DNA get into our cell? You know, DNA is a really linear or long structure, and how it gets into our cells is it packs itself. And the way it packs itself around these histones to start with. They're like little marbles or balls, and the DNA rolls around it. All right, and once it does that, then these balls, they get tightly packed together, all right, around the histones, and then the histones get closer and closer and make what we call chromatin. All right, and then the chromatin are like long strands, and then they pack up together, and eventually they make the chromosome. All right, I hope this gives you a brief overview of what DNA replication is, and I will talk to you soon.